All right, guys, I'll try and get through it in a timely fashion, just so we can all get to what we have to get to starting off the year here. Well, I just wanted to focus on kidney transplant because that's what I'm doing a fellowship in next year. So I thought it would be appropriate to get as much knowledge on the subject as I can. And I've done previous rounds on this as well, so I'll try not to go over what's previously been done. So I want to briefly review standard kidney donation practices. I want an overview of some of the previously discussed topics. Some have relevance to the presentation, and others just have some updates that I thought are worthwhile. I want to describe the concept of reverse transplant tourism, global paired exchange. I want to outline the potential benefits of dual kidney transplantation from marginal donors. And I want to review the history and current advances in kidney xenotransplantation with a focus on genomic engineering. So we all know the problem, and it's only getting worse in time. With all the transplants that we do here, you would think that we'd be putting a dent into the wait list, but never fear, it's growing every day. So there's US data is the top graph showing that wait list just going off into the sky with the rate of kidney transplantation staying pretty stagnant over the years. Below is recent Canadian data showing that our waiting list in Canada as a whole has stayed pretty stable, but in BC you'll see a different story. And the one number to always look at, may be hard to see, but deaths while on the waiting list. There's still 67 people in Canada dying on the waiting list, and most likely it's from cardiovascular causes. But you wonder if they had earlier access to transplantation, if they wouldn't have a different outcome. So kidney donation in BC. It's worth recapping this because it's the basis for what we all know. There we go. Basically, these are terms we should all know. Living donor. We do a lot of living donor transplantations. These are healthy people. The kidney gets out of the donor and into the recipient very quickly, and we expect pretty much immediate graft function from it, and we expect good long-term survival of the graft in most circumstances. Standard criteria donors. These also are healthy people who met an unfortunate demise before their time. They're often less than 60 years old, and typically it's brain death that causes their death. Extended criteria donor is something that we'll touch on a little bit again later in this talk. That's an important one to know about, and it's definitely an area of research, and that's where the marginal donors come into play. Donation after cardiac death is also another topic that we'll touch on a bit later. And as you can see, that bottom line is just signifying how grafts do once they go into people. Living donors do better than standard criteria donors, do better than donation after cardiac death, and so on. So kidney transplant in BC, there's two main techniques I can think of. We do a single kidney transplant into the iliac fossa versus a pediatric en bloc transplant, which I've had a chance to do here as well, which was a very interesting procedure. And then, of course, we do kidney pancreas transplants, occasionally kidney liver transplants, and occasionally kidney heart transplants. But as far as I'm aware, we don't do dual kidney transplantations from adult donors at this time. BC statistics, worth going over. They're updated every year because Transplant BC maintains such an excellent website and database. The number of kidney transplants in 2015 sort of blew my mind when I saw it. 271 transplants was done in 2015. That is just a huge number for the workload of the people that are doing. And to date so far, what are we at? About 190 transplants right now. So I don't think we're on pace to do what we did last year, but we're pretty close. So it's pretty impressive, the work that's being done here. Number of patients on the waiting list. I was really surprised at this, 492 patients. And last year I gave this talk and there was 361. So in BC we're having more people get on this, this list, which surprising with the volume going up as it is. So I just wanted to touch on three previously reviewed topics, and I'll try and give a different flair to them, sort of. So this was a review done in 2016. Samir did a really nice review in 2013 on small renal tumors for transplantation. And basically it's back table preparation. Small renal masses can be found from deceased donors, and they can be excised and decided to implant it, or there's many case reports now coming out of living donors having nephrectomies, back table explantation of the tumor, and then implantation with excellent graft survival. So this was just a review. There were 133 patients in total over a 10-year span. It was a short follow-up in the literature, maximum 6 years. But all the patients really and truly had good graft function. There was one recurrence of tumor. It was localized, and it was just conservatively managed at the 6-year follow-up. And complication rate overall is very low with these, very low rate of urine leaks. So conclusion, it's something that we can consider if we so desire. Health policy reform, this is a thing that I just touched on about a year ago, and there's an update on it. That's why I thought it was worthwhile sharing today. A year ago, people from the community, lay people and experts in the field met to have a kidney transplant summit here in Vancouver, and they asked two main questions. They asked, should BC adopt legislation that assumes everyone is an organ donor? And there was a jury that sat, and they did this very professionally, and they said, yes, we should, although this isn't the lawmakers. This is just a group that got together and says, yes, I think it's a good idea to have presumed consent, and the public seem to be in favor of that. Second one, should BC provide incentives? You know, we know that living kidney donation is a huge financial barrier to the people who go through it, and basically they said no. Kidney donation is an altruistic gift. It's a principle that we stand by, and it should not be financially compensated for.
How large is the financial burden on the donor on average?
I think it depends where they travel from, and I think there is some support to help out with that, but it's variable for sure.
It starts with the loss of income and then transportation. And that's the major one.
Housing, transportation, income, yep. Yep. So paired exchange, Dr. Gourlay did a really nice review on this last year as well. And paired exchange, I wanted to present it last as my review of topics, just because I'm going to go into the global paired exchange after this. We know paired exchange, we use that when we have a person with end-stage renal disease who has an incompatible living donor willing to donate who's healthy, and they can enter into a paired exchange whereby they go find other incompatible matches across Canada, and they swap organs, and a nice domino effect happens where a bunch of people get transplants who are more well-suited to their immunologic needs. So we know the basics of it, and we won't go into too much detail with it. So reverse transplant tourism, global paired exchange. I first heard about this at the AUA this last year, and it's an idea that was really pioneered by this guy named Michael Reese out of University of Toledo. And I think it's probably, they've done one of these reverse transplant tourism exchanges, and that was done in January of 2015, which I'll talk about in a sec, and there's been none done since. So I think there's definitely some ethical issues going on and some international constraints as I go through this topic for you to think about. So definitions, transplant tourism, that's travel to the developing world. If you're wealthy enough, you can give some money and you can get a kidney. It's a one-way flow of organs, which is really going against the tenets of what we believe in North America for how kidney transplant and kidney exchange should go. And there's potential risks to both the recipient and to the donor from these things, and they're well documented in the literature. Reverse transplant tourism, the idea by Michael Reese is trying to totally turn this idea of transplant tourism on its head. He has this idea of international partnerships. He wants everyone to benefit from it. He wants no non-transplant related cash incentives, but there could be transplant related cash incentives in his plan. And he wants the flow out of each country of organs to match the inflow, something different than transplant tourism. So the concept. It's a United States concept, and we'll focus on there. And the terminology that I use is Michael Reese terminology, which I probably wouldn't use the same one, and I'll talk about that in a sec. In the US, there's insured patients with a willing, incompatible donor. Now, some of these people aren't suitable for paired exchanges nationally, whether or not they have a high panel reactive antibody panel, or whether they're a blood group O recipient, which is somewhat tough to match from what I've been reading. just because O donors are universal donors and they can pretty much donate to anybody, whereas O recipients can only receive from O donors. So that's the biggest problem with that whole match. So in the developing world, so Michael Rees uses this term developing world, but a lot of the places I wouldn't consider developing that we talk about, he talks about Mexico, he talks about Philippines, I'd consider them maybe not as westernized as our world, but they're not what I would consider a developing world per se. But in those places, there are some poor people who are non-insured, who don't have access to health care, and don't have enough money to pay for the care that they should so need. Obviously, both of these cases present barriers to move the transplant forward, and it's neat that somebody's thinking about a way to try and solve this. So the keys. For reverse transplant tourism to work, there needs to be a good organization to identify patients from foreign countries with financial constraints to transplantation. So, I mean, you need a good infrastructure in that foreign country, and you need a good infrastructure in that place where you want this to occur. You need to partner with countries with enough of that infrastructure to ensure good ongoing transplant care and make sure they have the expertise to care for those individuals. And of course, you need to be able to fund this whole thing somehow, and where that money could come from is questionable. So the goals of healthcare reform, I thought those were worthwhile going over because reverse transplant tourism sort of hits on all of them. You need to reduce costs. We know 1 year of hemodialysis is about $90,000. A kidney transplanted person, it's about $33,000 a year. Most of that's upfront cost with the surgery, and the immunosuppressive costs ongoing are a little less than that. 5-year savings estimated in one study was $275,000 to transplant a person rather than be on dialysis. So Michael Rees asked the question, can a portion of health savings be used to fund reverse transplant tourism? It's not a bad question, because if you're saving so much money, that should go somewhere rather than pockets, potentially. Or maybe it should go pockets, because healthcare is getting more expensive in time. We want to improve quality. Well, if we're matching people better, that's going to improve quality. And if we're getting people transplants quicker, that'll also improve quality. And we want to increase access. And it's increasing access to, I guess, the developing world and also to those people at home. And it also takes people off the deceased donor wait list if we can get them transplanted. So, reverse transplant tourism in action, you need an organization. That organization identified a man from the Philippines with a compatible donor. So that's something interesting. This donor was totally compatible in our society. They would have gone ahead just with each other for this donation. But they didn't have enough money to undergo transplantation. And even this organization paid for some dialysis for this gentleman, Jose, before he actually could get the transplant. He was a blood group A, no panel reactive antibodies, and his wife was very, very healthy, and she was a universal donor. So reverse transplant tourism happened at, there's Michael Rees, he looks sort of like a mad scientist there, but he's, I met him, he's a very nice guy. University of Toledo, January 2015. There was a non-simultaneous extended altruistic donor chain. So what that means is there's an altruistic donor who starts off this chain, and it just continues on and on and on and on and on for as long as it can possibly go. And they try not to take that person off the deceased donor wait list at the end of it, if they can help it. But inevitably it will happen at one point. So this started with a US altruistic donor who wasn't a match for anyone in the US pool, surprisingly enough. This US donor was a match for Jose. And Christine, everyone wants her to donate to him because it's a good situation. So to date, the chain has resulted in 11 kidney transplants, 10 US citizens, and 1 is Jose. And that's them in the hospital. It was on the news. So the cost for Jose and Christine to get this all done, $150,000. That's travel accommodations. That's the dialysis before the transplant. That's the surgical care here or in the United States. And that's post-transplant care, which is that $50,000 number below. So they get a one-time fee of $50,000. And basically that's to pay for the immunosuppression and also post-operative care for both donor and recipient. And it's estimated in the Philippines that immunosuppression for the year is about $6,000 to $8,000. So the numbers don't add up for lifelong immunosuppression there based on this one go-around. So the ethics of this is quite questionable, I think. in that you're giving one thing to an American citizen, but this person who's come over and has benefited 10 American citizens is not getting that same treatment of lifelong immunosuppression that they would here. So it's something to consider anyways. And maybe that's one reason why it hasn't picked up steam, because there's a lot of merit to this to some extent. Basically, they estimated that they've saved over $3 million because of this gift that Christine gave and Jose received. So let's change gears here. Dual kidney transplantation is something that interested me because at Wake Forest, where I've accepted my fellowship, they're into this, and that's one reason that they get their numbers up, is they take these marginal donors that nobody else wants and transplants them. So I wanted to look into it a little bit just for my own benefit, but I think it's interesting as well. So there's two conflicting concepts about dual kidney transplants. One is that kidneys with a small nephron mass, they're going to undergo hypofiltration and glomerular hypertension, and this will lead to progressive renal failure over time. So the idea of a simultaneous transplant is to increase that nephron mass and maybe preserve kidney function for longer. The second is sort of against this, saying marginal kidneys have a functional reserve only that you can detect after the transplant. So what this is saying is we don't really know how they're going to work until they're in there and until we see them going. And it is true that GFR often increases post-transplant. So dual kidney transplant is redundant because potentially you could give those kidneys to two people rather than the one. So what's the definition of a marginal donor? I don't know. I tried to research it. I tried to figure out what the definition of a marginal donor is, and it's not clear from the literature. There's no real good definition of what it is. I definitely know that there's organs discarded all the time in Canada and the US, and they're discarded for very, very good reasons. There's no question about it. But some of those discarded, you have to wonder, is there a way that we could use them somehow to help keep access to kidneys available? So I just put extended criteria donors up there just to give you an idea of, these are the ones we think are the real marginal kidneys. But then can we go even more marginal than that? Age greater than 70 with or without risk factors for kidney disease, diabetes, hypertension. Age 60 to 70 with those risk factors, or a creatinine clearance less than 50. Those are some things that some people would consider marginal organs. And then there's also tools to sort of predict what marginal organs are, but they're not perfect like anything. So this is the Kidney Donor Profile Index. Basically, it basically creates a relative risk of kidney graft failure compared to the median kidney donor from the last year. So basically what that means is, it's hard for me to wrap my head around that. It means if you have an 85% KDPI, it means that 85% of donors have a better quality kidney than the one that you're going to be getting. And I think in some states they actually publish this to the recipients so they can accept or deny kidneys based on the KDPI if they're knowledgeable about this. Basically it's just a bunch of clinical and laboratory factors that go into this. But there is some evidence there about graft half-lives, and you can see if your KDPI is above 85%, your kidney graft median life is just over 5 years, which is not that good. So there's this Ramuzi score, which I'll just briefly touch on. Basically, it's a pathologic analysis, and it's a preemptive biopsy that's analyzed before the transplant goes in. And there's some correlation with nephron mass on this biopsy and a score of 0 to 3. Each of those 4 categories get a score of 0 to 3. And if you do score 0 to 3 total, then they say that kidney's good enough for a single kidney transplant. If you score 4, there's some authors that say you can transplant that as a signal, and some say you should do a dual kidney transplant. If you're a 5 or 6, it's a dual kidney transplant, and greater than 6, you should just discard that. But the overall validity of this particular scoring system is not that great. So dual kidney transplants from adult marginal donors have successfully expanded the limited deceased donor. So this is a study out of Wake Forest here. And I just wanted to touch on it briefly. Most of these studies I just want to touch on briefly, and they're all retrospective reviews, which is a limitation of the data for sure. But over a 12-year period, they did 72 dual kidney transplants, and they're doing more nowadays than they were back in the day. The creatinine clearance of their donors was less than 65 on average, and that was their target to go with the dual kidney transplant. And then they wanted to compare outcomes between the standard criteria donors that they transplanted and these dual kidney transplants. So basically, the 5-year graft survival, they estimated to be 84% in the dual kidney transplants, which is pretty comparable. I think standard criteria donors are probably 90 to 95% in most places.
I'm just wondering, Dave, are the potential recipients of these marginal donor kidneys also marginal recipients, or is it just the standard wait list?
No, it's generally speaking, it's done just like in Canada, where it's age matched. So a lot of these people are people who have been waiting for kidneys longer. They have more comorbidities, and you may not expect kidneys to do as well in them to begin with, just because of their comorbidities. So no, it's totally good. I left it out because I had to zip through some stuff. But no, it's a really good question. But essentially, basically, they found that their dual kidney transplant did better than the extended criteria transplants, and did comparable to the standard criteria donors, although a little worse overall.
Are the grafts broken down into a type of renal disease anywhere? I didn't see that.
No, like for the recipients,
So
what type of renal disease do they have?
GFR is the key.
For this study, yeah.
We'll do a diabetic kidney, nephrosclerotic kidney, whatever.
I think there was more that went into it than that. Definitely, if the comorbidities are mounting, sometimes they say, in a lot of places in the States, they do preemptive biopsies. And if it looks like they have a terrible Ramusi score, then they'll just discard those kidneys. So for all these ones where they have multiple comorbidities, that's probably the role for that preemptive biopsy more so than other things. That's a good question. So another study. This study is basically another retrospective review. This was in donation after cardiac death patients greater than 70 years of age. So that's a pretty high risk group, pretty marginal group overall. They used Ramusi scores to guide their transplants, and they did 12 dual ones with Ramusi scores about 4.5, and they basically found that all of the kidneys that they transplanted in this particular study did about as well as each other but again, follow-up is very short in this study. It was just about 2 years. Again, last one I'll just mention here. Another study looking at dual kidney transplant, and again, the results are very similar from these marginal donors. People do quite well with it, and pathologic stratification has potential value, although other studies would say pathologic stratification does not have value. Last one here is middle and long-term outcomes of dual kidney transplant. This is an Italian study, and just to say their 5-year survival, graft survival was 90% in this particular study, and their complication rates were very comparable to standard criteria donors, which is that graph on that right-hand side at the bottom.
You're only talking about adults, not pediatric.
I didn't. I had a slide on pediatric tool on block, but I cut it. Yeah, I'm just talking about adults. Yeah. Pediatric ones, it was really interesting. I don't want to digress too much, but their success rate was quite a bit lower than what I was seeing with the adults, likely because of the small vasculature, I would imagine. And I didn't want to do anything that we were doing here as well. I wanted to try and do something different. So dual kidney transplant, after reviewing it, I think it's something we should consider. I think it's, I'd be curious to know if it's something we've thought of. I'm sure it's been done here before.
One of the considerations with that is you're in the adult ones, you're losing it by half. And so if you're looking at the longevity of these kidneys being equivalent to the extended criteria donor, so you're getting 70 years of it. So who are you putting these kidneys into? Are you putting them into a 40-year-old who 10 years from now needs both a transplant nephrectomy and a retransplant? Or are you putting them into the 75-year-old who, if you get them to retransplanting following transplant nephrectomy is a much different operation, much different outcomes.
Yeah.
So you in essence, you're burning both bridges as you're putting this one graft in. So I think patient selection and your plan when this fails is the important consideration, not the initial graft. It's what's going to happen when this fails, because they all do.
Yep, for sure. Until xenotransplantation, which is coming, those are not going to fail. But anyways, I totally agree with you. It has to be the right recipient for this. There is a lot of literature on doing dual same side, but it's from what I could tell, I think it's increased DVTs and increased thrombotic events when you do it on the same side, just a little bit. And but graft function's the same, comfort's the same, and you could still potentially preserve one side. But I totally agree. I think this is reserved for an older patient who may be down on the list for various reasons, and the kidneys are marginal and they're willing to take that risk to get something sooner. But maybe worth a talk to figure out who those patients are. Graft function complications, patient survival appear acceptable. It's really hard to know what a marginal kidney is. How far outside of the extended criteria donor are you willing to go to actually transplant these? No one knows that question. And there's no prospective trials, so it'd be interesting to see because the wait list is an issue. So xenotransplantation, the last topic I just want to touch on, which is I'm amazed how much xenotransplantation research is going on around North America and around the world. It's quite startling. It's something that certainly we don't hear about every day. But this guy named Thomas Starzl, he's I guess the godfather of xenotransplantation at the University of Pittsburgh, which is a massive transplant center. He had this quote, and I think it's a great quote. History tells us that procedures which were inconceivable yesterday are barely achievable today, often become routine tomorrow. And
I think he's a grandfather
Is he?
of transplantation in general.
Oh, is he?
He's the guy that Marty did his fellowship
At Hopkins?
with. You know what, was it when we were calling Denver? He's in Colorado.
Whatever he is, he has a great quote. That's a really good quote.
Pointed, he's the Bach and Beethoven of transplantation.
It's an inspiring quote, really, because it wants you to push the limits and to see what else you can do. So the WHO definition, just to review it, is it's pretty obvious what it is: transplantation of living cells, tissue, or organs from one species to another. In the realm of transplantation, this is pretty exciting. If we could figure out how to make it work, there'd be no more deceased donors, there'd be no more living donors, there'd just be endless supply of xenotransplants if we can get over the medical, legal, and ethical considerations. So what are the barriers to xenotransplantation? Well, the human immune response is definitely something that we always have to take into account. There's physiological incompatibilities between pigs and humans. And I'll let you know why I say pigs here rather than other animals. One is their lifespan. A second one is there's been a significant proteinuria associated with kidneys going into different species, and it's not exactly clear why, but it seems like with better immunosuppression, that's actually self-resolving, so it's hard to know if that's as big of a problem as we think. And then there's the theoretical risk of transmitting zoonotic pathogens from pigs to humans. Although there's no documented evidence of it, pigs do have these things called porcine endogenous retroviruses, and I guess humans have these very same things, and it's something that certainly to get approval, it's an issue that needs to be dealt with. So kidney xenotransplantation, there's a long history of kidney xenotransplantation dating back to the early 1900s from this particular graft. A patient was given rabbit kidney and lasted 16 days, and then a few other things were tried, and they didn't last very long. In the 1960s, xenotransplantation got a bit of a renewal. 13 patients received a chimpanzee kidney, and one even lasted 9 months, surprisingly, but most didn't last very long at all. And of course, they died because of rejection or infection. And then baboon kidneys went into 6 patients, also in the 60s, and again, their survival was quite dismal. So after seeing humans die quickly after an intervention like this, this was put on ice. And in the 1980s, somebody thought we should start this again, and they developed a pig to non-human primate model. And the biggest question is, why pigs? Is the biggest question. And pigs are what we use in our animal lab all the time to do our surgical skills on. And why we do that, I never really realized that before this, but there is a lot of pigs in this world. They're breeders. They get full grown fast. They have organs about the same size as us, and the public opinion of it's generally more in favor than doing something like non-human primates, which look like us. And if you believe in evolution or creation, I'm not going there. Whichever you believe in, maybe we evolved from them at some point, or we were distant ancestors in the past. Kidney xenotransplantation. This is a really interesting chart, I think, because this is the mean survival of transplants going into non-human primates. And if you look in 2015, there's a gigantic jump up in the survivability of that graft in the kidney. So something must have changed. It's only one. No, but there's another graph that has the bar going up. Why the sudden jump? Because there was a case of a non-human primate living 133 days. So it's all about genomic engineering. And I'm not a biomedical engineer, and I'm not going to go into super gritty details of it because I'd mess it up. But the basics, I think, is worth knowing because it's really cutting-edge stuff. It's really exciting. Genomic engineering, it's a process whereby researchers can introduce a modification into a genome. Sounds pretty simple. Remove, insert, replace, disrupt. Typically, this is done at the embryonic level, although there is gene-targeted therapy that can be done in people as they go older as well. Basically, the idea with most of this genomic engineering for kidney xenotransplantation is heritable changes to genomes that can be passed through generations. So some past attempts at genomic engineering, which were very successful, but were limited by the length of time and the expense, were these zinc finger nucleases and these talons. Those are something you may hear about, but with this new kid on the block, this, I call it CRISPR Cas9, but I'm not sure if that's actually how you're supposed to say it. It is exciting stuff, and this is coinciding with that big jump in the survival of that particular non-human primate. So what's the story with CRISPR? Well, it's a real fascinating success of the world scientific community coming together. It starts with the discovery, and it goes on to just honing in on what CRISPR means to be able to being able to splice mammalian cell genomes and put precise segments of DNA in specific locations in the gene. It's fascinating stuff. So what is it? A little bit more detail. Well, it was found as a mechanism of adaptive immunity in bacteria located on the plasmid. It was essentially set up to defend against viruses that inject their DNA into the cell. This plasmid can essentially, with this protein called Cas9, chop up that viral strand. And once that viral strand's chopped up, it's essentially inactive from what I've been reading, and they can incorporate it into their own DNA so they can recognize that virus for future times. So it's a mechanism to chop up some foreign DNA and incorporate it into your own genome at specific locations to be able to recognize them, to be able to attack again in the future. And some very smart people have figured out a way to harness that to modify the genome. So CRISPR is clustered, regularly interspaced, short palindromic repeats. We will refer to it as CRISPR from here on out. And Cas proteins are just CRISPR-associated proteins, and they're vital. to being able to target specific sequences of the DNA. This is a nice simple diagram of how it works. It's pretty wordy, but essentially the gist of it is that there's 4 main steps. One is you need a specific single-stranded RNA. That's your targeting domain, basically. You need to attach that to a protein, the Cas9 protein. The Cas9 protein will find a specific segment of DNA on the target DNA strand you want to disrupt. That Cas9 protein's like a pair of scissors that can cut both strands of the DNA. And then there's different techniques they can do to sew things back up, put new things in, or frame shift things to get different changes. Why are we so excited about this? And you can see that the number of publications about CRISPR-associated changes has gone up over the last couple of years. The patents has gone way up over the past couple of years. I mean, it's pretty exciting to be able to perform gene editing in mammalian cells. But the zinc proteins in the talons could do this as well. This is much more exciting because it's easier, quicker, cheaper, and it's pretty much available to any lab that wants to get involved with it. And it's also exciting because if you change a short segment of RNA, you can get a different site in the genome. So there can be a high degree of precision, but there can also be a high degree of error if you don't change it right. So what are the problems with it? Well, this off-target effect is definitely a well-documented problem with it, and meaning if you do have specific plans to get a certain genome, many of the cell lines that you create will not have that particular genomic effect. There's also regulation ethics issues. There's people in different countries doing things that certain people in some countries don't think is totally cool. Some Chinese scientists, they made changes to human embryos, and people didn't like that, even though these were non-functional embryos that couldn't be implanted. There was also a case of American researchers aerosolizing the CRISPR-Cas genomic effect in hopes of treating lung cancer in a animal model. And they had everything closed off, but there was really concerns about this being aerosolized because if it got out, what could this mean for anyone who would breathe it in, or even anything living like plants, insects, everything could be affected potentially. So there's huge regulation issues with this. Genomic engineering and xenotransplantation, there's many different organs and structures that are potential targets for this. The kidney is the one that we're interested in, and there's some exciting stuff there. So what genomic modifications are needed? Well, basically we need to figure out the barriers to xenotransplantation, and success. It's likely going to happen because of expression of multiple transgenes, so genes that we put in. Endogenous gene knockouts, genes that are troublesome to the immune system, we got to get rid of. However, we got to get rid of them in a way that doesn't endanger the livelihood of that animal that we're knocking them out of. And of course, immunosuppression is a standard of transplantation, but I mean, the dream of genomic engineering and xenotransplantation would be no need for immunosuppression, although as of now, it's a pipe dream. So on the left side, you have the unprotected pig endothelium. This is with no genetic modifications. HAR is hyperacute rejection, which was the primary mechanism for why these grafts failed so quickly. You have the acute vascular rejection in the middle, and you have chronic rejection in the bottom. And on the right side, some ideas for some genomic modifications that can happen, and I'll talk about those, some of them very briefly. So the barrier, hyperacute rejection, how can we get around this? Well, this came out of, initially there was a paper out of the University of Pittsburgh in '92, which isn't this one, but it identified this surface protein in pigs and other non-primates that is basically 1,3-galactose, and the enzyme coding for that is the 1,3-galactotransferase. Basically, there's been genetic modifications to knock that gene out, and now there's a whole line of pigs right now without that gene, and indeed they do live longer and they do avoid hyperacute rejection just by knocking out that one gene alone. And then you can see there the survival was up to 83 days. Acute vascular rejection, human tissue factor has basically been added into the pig genome. And again, it does, it avoids that acute vascular rejection where you have increased clotting time and you have decreased thrombin clots that are represented in that graph on the right. The production and characterization for chronic rejection, there's this immunoglobulin CTLA4, which is basically an inactivator of T cell activity. Pigs have been engineered with this, and it certainly does decrease the T cell activity and high expression. However, the big problem with it is it significantly compromises the immune status of the pig. So you're having pigs that are dying with this modification as it is. But it could be something that they're going to try and figure out a way around that in the future. And this is the zoonotic pathogens, genome-wide inactivation of porcine endogenous retroviruses. Whoa. This is really exciting or scary, whichever way you want to look at it. Basically, the CRISPR technology has been used to inactivate 62 retroviruses in the pig genome at once, which is the largest to date. It's huge. So what do we take from this? Longest life supporting xenotransplant to date, 132 days. 6 modified genes in this particular one. More coming down because CRISPR-Cas9 is going to be, they say it's the revolution. We'll see. Questions, and thanks to Dr. Bond.